Jetzt können wir loslegen.
This is the English version of a lecture at the Institute of Public Health of Heidelberg University on the 16th of February 2011 by Henk Tennekes, entitled Toxicity as Function of Exposure Time, Risk Profile of Neonicotinoids, Insecticides and the Consequences. Let me first of all introduce myself. I graduated from the Agricultural University of Wageningen in 1974 and performed my PhD work at Show Research in Sittingbourne in the UK. I worked at the German Cancer Research Center in Heidelberg from 1980 to 1985, where Hermann Druckrey became my mentor. And during our last meeting, Druckrey, who was a renowned pharmacologist and cancer researcher, provided a copy of a book that he published in 1949, the result of a collaboration with the mathematician Karl Kupfmüller on theoretical principles of dose-response relationships in toxicology and pharmacology. Known at that time was Haber's rule, the product of concentration and exposure time produces the same biological effect. Thus, the rule states that doubling the concentration will halve the time up to the same effect. In a linear plot, the relationship appears as a hyperbole, the red curve on the right-hand side, on logarithmic coordinates as a straight line. Druckrey was interested in this relationship because he'd shown in rats that it described the liver carcinogenicity of 4-dimethyl aminoazobenzene, also known as butter yellow. He noted that CT poisons, as he described them, were characterized by irreversible binding to cell constituents, or were inducing irreversible functional changes in them. Druckrey and Kupfmüller then used Paul Ehrlich's theorem that a pharmacological effect results from a chemical or physical reaction of a poison with specific receptors in reaction kinetic terms. But first, they described the processes that determine the complex relationship between dose and effect. The first process is absorption, which determines the relationship between dose and the concentration of the poison at the site of action and its time course in various parts of the body. The second process comprises the chemical reaction of the poison with specific receptors and is about the relationship between the concentration of the poison at the site of action and receptor binding. And receptor binding will determine the third process, the actual biological effect. And here are the reaction kinetics of a bimolecular reaction of butter yellow with specific receptors. And here you see this is the velocity of association, and here the velocity of dissociation, where R is the initial concentration of specific receptors that butter yellow reacts with. CR is the concentration of bound receptors. C, the mean but yellow concentration at the site of action, induced by daily but yellow doses. And K is a reaction constant for association, and TR, the time constant for dissociation. Now, by a few very simple assumptions, Drucre and Kupfermüller demonstrate that this equation 1 reduces to Haber's rule, which is equation 4. First of all, they assume that there is no dissociation, i.e. that the time constant for dissociation approaches infinity, and this reduces equation 1 to equation 2. Then they assume that the concentration of bound receptors remains very much smaller than the initial concentration of specific receptors, which means that the initial concentration of specific receptors will remain approximately the same, which leads to the simplification of equation 2 to equation 3. And if then you assume that the mean but yellow concentration at the site of action is constant, then integration leads to equation 4, which is Haber's rule. So with very simple reaction kinetics, Drucre and Kupfermüller showed that Haber's rule was consistent with irreversible receptor binding, but it assumed proportionality between the concentration of bound receptors and the effect, and this may not always be the case. The reversibility of receptor binding can have the same significance for dose response characteristics as the reversibility of the effect. An effect will depend on the concentration of the poison at the site of action only when both receptor binding and the effect are quickly reversible. That is curve 1, the concentration of the poison at the site of action. If receptor binding is irreversible, the effect will be determined by the integral of compound concentration at the site of action over time. This is exemplified by curve 3. Where curve 1 represents the concentration of the poison at the site of action. Now then, if both receptor binding and the effect are irreversible, the effect will be determined by the double integral of compound concentration over time. This is exemplified by curve 4. And in this case, the effect is reinforced by exposure time. 15 years later, Drucre and his co-workers demonstrated, and in 1963, Drucre and his co-workers showed a reinforcement of an effect in a liver carcinogenicity study in rats with diethyl nitrosamine. In a log-log plot, so a plot on logarithmic coordinates, the relationship between the daily dose of diethyl nitrosamine and the median tumor induction time was a straight line. And this appears to be a general appears to have general applicability, because in a photocarcinogenicity study in hairless mice, it was also shown that the time up to the appearance of skin tumors was linearly related to the daily dose of ultraviolet light. This curve is the curve for ultraviolet B light, and this for ultraviolet A1 light. Now, the essence of this relationship was that the carcinogenic effect is reinforced by exposure time. The lower the daily dose, the longer it takes before cancer appears. However, the lower the dose, the daily dose, the lower the total dose required for a carcinogenic effect. And in fact, a safe dose cannot be defined for carcinogens. A single hit may suffice to induce cancer, and this can be shown with the retinoblastoma protein, which is a tumor suppressor protein that is dysfunctional in many types of cancer. A mutated retinoblastoma gene can be inherited, and the mutated gene is recessive. So if a cell sustains only one mutation in the other retinoblastoma gene, then all the retinoblastoma proteins in that cell would be ineffective. Now then, let's come to the toxicity of neonicotinoid insecticides to arthropods. The toxicity of imidacloprid and thiacloprid to neonicotinoid insecticides to arthropods shows very much the same dose-response relationship as we've seen for chemical carcinogens. In a log-log plot, the relationship appears as a straight line. The concentration here of the toxicant and the time up to a lethal effect on the y-axis. And as we've seen with carcinogens, the latent period is inversely proportional to concentration. It takes a long time before, at a very low dose, you see a lethal effect. At a high dose, you see it much earlier. But the effect is reinforced by exposure time. The total dose required for a lethal effect is much lower at a low daily dose, at a low concentration than at a high concentration. So very much the same characteristics as we've seen for chemical carcinogens. Now, the mechanism of action of neonicotinoid insecticides also shares similarities with chemical carcinogens. Imidacloprid, the most widely used neonicotinoid insecticide, was the first insecticide whose mode of action was found to derive from almost complete and virtually irreversible blockage of postsynaptic nicotinic acetylcholine receptors in the central nervous system of insects. And there is an additional problem with imidacloprid. The compound is persistent and mobile in soil and prone to leaching. Imidacloprid has potential to leach to groundwater. And in addition, the high solubility and mobility are concerns for transport to surface water by dissolved runoff. And imidacloprid is very persistent in soil. The shortest half-life was 170 days in turf-covered soil in Georgia, but in Minnesota cornfield soil, the imidacloprid concentration did not decline for 1 year after treatment. And here you see on the left-hand side the applications of imidacloprid in the Netherlands. Yellow is maize, brown potatoes, blue sugar beets, black is greenhouse crops, green orchards, and red flower bowls. And the use of imidacloprid in these branches of agriculture is associated with surface water contamination, which exceeds, in many cases, the maximum tolerated risk limit. And in some places in the Netherlands, the contamination of surface water with imidacloprid is massive. In Waddinxveen, the limit is exceeded nearly 1,000 times. In Rijnsburg, nearly 2,000 times. And in Noordwijk, nearly 5,000 times. Now then, we now know from recent studies that have been conducted in Western East European countries that agricultural intensification has a negative effect on wild plant, carabid and bird species diversity. And the important thing is that of the 13 components that were measured, the use of insecticides and fungicides had consistent negative effects on biodiversity. And insecticides also reduced the biological control potential. And what we see in the Netherlands is a decline of farmland birds. Here you see data for the corn bunting, the blue line, and the ortolan bunting since the introduction of neonicotinoids in the early 1990s. In fact, these two bird species are now extinct. The wingchat is no longer seen on Dutch farmland. The skylark is in massive decline. The population of the grey partridge is also in steep decline. And the common pheasant is also declining in Holland. Now, this surface water contamination with imidacloprid correlates with a reduced Diptera abundance. So a reduced abundance of flies and midges. And I've shown this again here in units of MTR units. So units of the limit, the maximum tolerated risk limit. And you see that when the surface water contamination is only slightly above the MTR, 1 to 5 times, you already see clear changes in diptera abundance. And you remember that in Rheinsburg this limit was exceeded nearly 2,000 times, and in Noordwijk nearly 5,000 times. Well, the bottom line is that there are no longer midges and flies in those areas. Again, here on the left-hand side, the applications of imidacloprid, and on the right-hand side, the groundwater levels in the Netherlands. And as you can see, in large parts of the country, the groundwater level is below sea level. So Holland is extremely susceptible to leaching. And we assume that imidacloprid contamination of Dutch groundwater may be widespread. What we see is a strong decline of butterflies since the introduction of neonicotinoid insecticides. But also bird species in very different habitats are declining. Here's the black-tailed godwit, which breeds in wet meadows and is on the red list now. The willow tit breeds in mixed woodland and is in steep decline. The crested lark breeds in open areas with sparse vegetation, now extinct in Holland. The oyster catcher is one of the most characteristic birds of the North Sea coast and is projected to be extinct in Holland by 2020. The great reed warbler breeds in aquatic vegetation emerging from shallow standing water, is also endangered in Holland. The northern wheat ear breeds in coastal dunes and is nearly extinct in Holland. The tawny pipit breeding on heathland is now extinct in Holland. Associated with human altered settings is also declining in Holland. And the house sparrow, closely associated with human populations, is in steep decline. So the conclusions are that the mode of action of neonicotinoid insecticides derives from almost complete and virtually irreversible blockage of postsynaptic nicotinic acetylcholine receptors in the central nervous system of insects. The toxicity of neonicotinoids to arthropods is reinforced by exposure time. Their dose-response characteristics are strikingly similar to those of carcinogens. Therefore, there may not be a safe level of exposure. Imidacloprid is persistent and mobile in soil and is prone to leaching. The contamination of surface water with imidacloprid is massive in some parts of Holland. We know that of the 13 components of agricultural intensification. Only the use of insecticides and fungicides had consistent negative effects on biodiversity, wild plants, carabid and bird species. The surface water contamination with imidacloprid correlates with reduced diptera abundance, and there is a strong decline of butterflies since the introduction of neonicotinoid insecticides, and many invertebrate-dependent bird species in very different habitats are declining, and some are now extinct. Although we cannot claim at this stage that we've demonstrated causal relationships, these data are highly disturbing, and I decided to write a book to warn the general public about the systemic insecticides. We must address this issue. It could be a disaster in the making.